All right, well, welcome to microbiology. This video is going to do a review of the microscope and focusing skills. The first lab of the quarter, you'll use these prepared slides. So I'm using one of these. And just as a heads up about that, you see how each slide has a numbered home. And please make sure to always get your slide back in that numbered home. Okay, so the one that I'm using today is Enterobacter aerogenes. So you'll want to practice saying some of those terms. Enterobacter aerogenes. This is a gram-negative rod, which means it's going to look pink under the microscope. So I'm going to put the slide on the stage, and then I push on the stage clips that are right here. And I open it and slide the slide nice and tight in there. Then I let go of the stage clip, and I can hold that slide in place. Now, you always need to make sure that it's centered, especially with bacteria, because they're so small, there might be only a few of them in one area on the slide, and that's where you want to work on your focusing. So in order to center it, you can move the slide up and down with the top adjustment and side to side with the bottom adjustment. So I'm going to look for that pink blob, and sure enough, there it is. And now I have it centered. So you just want to make sure it's always centered before you do the hard work of trying to focus. The stage should be all the way down when you first get your microscope out, and that's the way you should put it away. But when you're ready to start work during lab, you need to raise that stage all the way up like this. Now you see how far I had to bring that up? If you didn't do that, you would be spending a long time trying to focus. So that's a really helpful tip for you to remember. Okay, now what I'm going to do is look through the oculars, and I'm going to just lower the stage a tiny bit to bring this into focus. If you have trouble adjusting these oculars for your eyes, use this ruler right here to measure the number of millimeters that are between your pupils, what's called the interpupillary distance. And let's say it ends up being 62 millimeters between your pupils. Then you're going to set it up at 62 like that, and you should be able to look into the microscope and it will be focused. There are further adjustments that can be done for focusing like this if you have one eye that sees much differently than another. That is something just by trial and error that you can work on. And then if you find that there's a measurement that works for you, remember that number and return to it the next lab. Okay, so for me, I'm going to look through the microscope, and I moved these oculars together so that I get one large circle. You don't want two circles. You want one large circle. For me, it looks like that's right about at 64 millimeters. Okay, so now I'm going to lower the stage till this comes into fine focus. And do you see I'm not moving it very much? And ta-da! I have Enterobacter aerogenes focused at low power. So since these microscopes are parfocal, I can turn it right to medium power, which is going to magnify the specimen about 100 times. And it's almost perfectly in focus, and just make a tiny adjustment there. Now the next thing I do is make sure that it's still centered, because as you go from lower to higher powers, your field of view will shrink. And so you want to make sure that it's very nicely centered. And remember, you use those mechanical stage directives. And most of you will have a pointer in your microscope, and make that pointer right exactly on what you want to see at high power. So sure enough, then when I turn to high dry, I should be able to see it. And I do. And at this point though, notice that I'm turning the fine focus, not the rough. This is the rough, this is the fine. I am turning the fine focus. The next adjustment I need to make is to turn the light up just a little bit at this point, because the higher the magnification, the more light that you will need. Okay, now the next step is fun. We're going to go to oil immersion. So I put the objectives between what we call high dry and oil immersion. See how it has that white line on it? Take the oil immersion fluid that is in your drawer, and I'm going to put 1 to 2 drops right on the sample. I'm going to use a Kim wipe to wipe my fingers so I don't get oil all over the other parts of the microscope. And I would appreciate it if you did that too. Okay, and so now I'm able to look at a higher magnification than many of you probably have ever used in a biology lab, so that's kind of exciting. What oil does is it makes the refractive index for light the same as the objective. It's as if the objective is touching the slide. You can't really have the objective touch the slide or you would damage the objective, but the oil helps to make that refractive index continuous from the bottom of the objective all the way to the specimen on the slide. Okay, so now I've got it focused at oil, but it's not perfect. So these are some things you'll need to remember to do. When you finally have it focused at oil, you need to lower the condenser. The condenser knob is right here, and as you lower it, I turned it down about half a turn. You'll notice right away that you've got to turn the light up. You're going to have to turn the light up now, and then do a tiny focusing, and that's going to increase your resolution. Just like it sounds, the condenser changes the way the light is condensed on the specimen, and it helps you see the contrast better, which can make it easier to look at your sample. Okay, so just to review, what we did is we put the slide on to the microscope using stage clamps. Then we brought the stage all the way up at low power and then lowered it just a tiny bit until things came into focus. We always made sure to center before going to the next higher power by using the mechanical stage directives. Each time we turned from one power to the other, we would make slight adjustments with the focus and also with the light. The trend is you have to turn the light up higher and higher as you go from low all the way to oil immersion. Then we put 1 to 2 drops of oil on and made some final adjustments there. It's very important to lower the condenser and turn up the light, and I will expect to see that you have done that when you're working.